ever just, stop and think about how sunlight hitting your eye becomes, well, color? Or how vibrations turn into a voice you recognize?
It's happening constantly, isn't it? This amazing connection.
Yeah, and we often don't even realize the incredible, complex stuff going on behind the scenes.
It's truly remarkable. And that seamless experience, it relies on these really intricate biological mechanisms detecting and interpreting a constant flood of information.
Which is exactly what we're diving into today.
Precisely.
We're exploring the fascinating world of sensation and perception. We've looked at a range of research to unpack how we detect and make sense of everything around us.
Mm-hmm.
And our mission for you, listening in, is to get a really clear, engaging handle on this whole process, from the physics of a light wave all the way to how your brain builds your reality.
And we'll cover the main players: sight, sound, taste, smell, touch.
And even the ones we don't think about as much, like balance, knowing where your limbs are, all that. But without getting bogged down in super technical jargon.
Sounds good. So where do we start?
Well, where does it all begin? The detection part.
Exactly. It starts with sensation. Think of it as the initial detection. We have these specialized nerve cells, sensory receptors, and they're the gatekeepers.
Gatekeepers, I like that.
Yeah, and each type is tuned to a very specific kind of stimulus. Some for light, others for sound waves, pressure, chemicals. You get the idea.
So they're like tiny detectors waiting for their specific signal.
Spot on. And when they get activated by that signal, that's the very beginning of sensation.
But it's not enough for them just to get, tickled by the environment, is it? There's got to be a translation step.
Absolutely crucial. That's called transduction. It's basically converting one form of energy into another.
Okay.
So the sensory stimulus, light energy, sound energy, whatever it is, gets converted by these receptors into electrical signals, action potentials. That's the language the nervous system speaks.
Okay. Like translating English into Spanish so someone else can understand.
Exactly like that. Translating the outside world's language into the brain's language.
Now, I think most of us learned about the five senses, right?
Yeah.
Sight, hearing, smell, taste, touch. But the sources suggest that's maybe a bit too simple.
Yeah, that's a common starting point, but it's definitely a limited view. Our sensory world is much richer.
Like what else?
Well, think about your sense of balance, keeping upright without thinking about it. That's the vestibular sense.
Right, yeah.
Or just knowing where your hand is right now, even with your eyes closed. That's proprioception and kinesthesia, body position, and movement awareness.
Huh. Never really thought of those as senses in the same way.
But they are. We also have specific systems for detecting pain, that's nociception, and for temperature, which is thermoception. So yeah, way more than five.
It's kind of mind-blowing how much our body is tracking all the time without us, consciously tuning in.
It really is.
So with all these senses constantly working, there must be a point where a signal is just too weak to notice, right?
Yes.
A lower limit.
Definitely. That brings us to the absolute threshold. It's defined as the minimum amount of stimulus energy needed for you to detect it about 50% of the time.
50%. Why not 100?
Well, it accounts for slight variations in attention and sensitivity.
Yeah.
But the point is, our senses can be incredibly sensitive at this threshold.
How sensitive are we talking?
Well, the classic examples are pretty amazing. Like, on a perfectly clear, dark night, you could theoretically see a single candle flame from about 30 miles away.
30 miles? Seriously? Wow.
Or in total silence, hear the tick of a watch from 20 feet.
That really puts it in perspective. Our systems are incredibly fine-tuned.
They really are.
Which makes me wonder about those, subliminal messages people talk about. Stuff below that threshold of awareness. Do they actually work? Can they influence us?
That's a really common question. And the research is interesting. In controlled lab settings, yes, our brains can process information presented below conscious awareness. But, and this is the important part, the evidence strongly suggests that in the real world, these subliminal messages have very, very little, if any, real effect on our behavior. You're not likely to be unconsciously swayed by a hidden message flashed in an ad.
Okay, that's somewhat reassuring. So we have the minimum needed to detect something at all. But what about noticing a change? Like if the TV volume goes up slightly, that feels different.
It is different. You've nailed a key concept there. That's the just noticeable difference, or JND, that's sometimes called the difference threshold.
JNT.
It's the smallest change in a stimulus that we can reliably detect. But here's the interesting part. It's not a fixed amount.
What do you mean?
It depends heavily on the intensity of the original stimulus. Think about your phone screen. In a completely dark movie theater, even a tiny bit brighter is super obvious, right?
Yeah, totally distracting.
But now imagine you're outside on a bright, sunny day, or in a brightly lit basketball arena. That same tiny increase in brightness, you probably wouldn't notice it at all.
Ah, right. Because the starting brightness is so much higher. It's all relative.
Exactly, it's relative. And this leads us to something called Weber's law.
Okay, Weber's law. What's that about?
It's a principle observed way back in the 1830s by Ernst Weber. He noticed that the just noticeable difference is a constant proportion or fraction of the original stimulus intensity.
A constant proportion. Okay, so give me an example.
Imagine holding a small weight, say 10 pounds. You might notice if someone adds another pound. But if you're already holding 100 pounds, adding just one more pound, you probably won't feel the difference. You'd need to add proportionally more, maybe 10 pounds in that case, to notice it.
Makes sense. The stronger the initial feeling, the bigger the change needs to be for us to pick up on it.
That's the core idea of Weber's law.
Okay, so that covers the basics of sensation detecting stuff, converting it, the limits, the fact we have more senses than we thought, and how we notice changes. Let's zoom in now. How about vision? It feels like our dominant sense, maybe.
For many people, yes. It's incredibly complex. Let's start with light itself. It travels in waves.
Right.
And two key properties of these waves determine what we see: amplitude and wavelength.
Amplitude and wavelength. Okay.
Amplitude is the height of the wave. Think of it like the intensity. Higher amplitude generally means we perceive the light as brighter.
Okay, height equals brightness. What about wavelength?
Wavelength is the distance between the peaks of the wave, and that's primarily what determines the color or hue that we see.
So short waves versus long waves give us different colors.
Exactly. And the light we can actually see is just a tiny, tiny slice of the much broader electromagnetic spectrum.
Ah, yeah, the spectrum. That includes like radio waves, X-rays.
All of that. Gamma rays, microwaves, infrared. Visible light is just this narrow band, roughly from 380 to 740 nanometers. That's it.
Wow, just a sliver. And other animals see different parts.
Oh, yeah. Bees, for instance, can see ultraviolet light, which is beyond our violet range.
Yeah.
And some snakes can detect infrared radiation, which we feel as heat but don't see.
Imagine seeing in UV. That'd be wild.
It would change your whole perception of a flower, for instance.
So, back to our visible slice. How do the wavelengths relate to the colors we know, like ROY, by?
It follows that order pretty well. Longer wavelengths around the 700 nanometer end we see as red. As the wavelength gets shorter, we move through orange, yellow, green, which is kind of the middle, then blue, indigo, and finally violet at the shortest end, around 400 nanometers.
Red is long, violet is short. Got it. Okay, so these light waves hit our eyes. Walk us through the eye itself. What are the main parts doing?
Okay, so first, light enters through the cornea. That's the clear, curved outer layer at the very front. It does a lot of the initial focusing.
Like the first lens element.
Sort of, yeah. Then the light passes through the pupil, that black hole in the center.
Which gets bigger or smaller, right?
Exactly. The iris, which is the colored part of your eye, controls the pupil size. It acts like the aperture on a camera, opening wider in dim light to let more light in, and shrinking in bright light to reduce the amount.
Cool. So cornea, pupil, then what?
After the pupil, the light hits the lens. This is another transparent structure, and its job is to fine-tune the focus, bending the light so it lands perfectly on the retina at the back of the eye.
The retina, that's where the light detection actually happens.
That's the light-sensitive lining, yes. And for really sharp, detailed vision, the light needs to be focused on a tiny central spot on the retina called the fovea.
Fovea. Okay.
The fovea is packed almost exclusively with one type of photoreceptor cell called cones. Cones are responsible for sharp detail and color vision, but they need relatively bright light to work well.
Cones for color and detail and good light? What about the rest of the retina?
The rest of the retina has cones too, but it's dominated by the other type of photoreceptor, rods. Rods are much, much more sensitive to light than cones.
Ah, so they handle low light vision.
Exactly. They're crucial for seeing in dim conditions. They're also really good at detecting movement, especially in our peripheral vision. But they don't provide much detail or color information.
Okay, so cones, sharp color vision, daytime. Rods, sensitive low light vision and motion detection, nighttime. That makes sense why colors look washed out in the dark.
Precisely. And issues with rods not working properly are often behind night blindness.
Right. So light hits the rods and cones. How does that signal get from the back of the eye to the brain?
Good question. The rods and cones connect to other neurons right there in the retina. Eventually, the signals converge onto retinal ganglion cells. The long tails or axons of these ganglion cells bundle together to form the optic nerve.
The big cable running out the back of the eye.
That's the one. It carries all the visual information away from the eye towards the brain. And interestingly, where the optic nerve leaves the eye, there are no rods or cones. That creates a blind spot in each eye.
But we don't notice a hole in our vision.
Nope, because the blind spots are in different places for each eye, and our brain cleverly fills in the missing information based on the surroundings. It just patches it over.
Our brains are pretty smart. So the optic nerve heads towards the brain. What happens next?
The optic nerves from both eyes meet underneath the brain at a structure called the optic chiasm. Think of it as a crossover point.
Crossover.
Yeah. Here, information from the right side of your visual field, what you see to your right, captured by both eyes, gets sent to the left hemisphere of your brain. And information from the left visual field goes to the right hemisphere.
Huh. So it swaps sides. Why?
It allows each hemisphere to get a complete picture of the opposite side of the visual world, which helps integrate information. From the optic chiasm, the signals travel through other relay points, like the thalamus, and finally arrive at the occipital lobe, right at the back of the brain. That's the primary visual processing center.
Occipital lobe. Okay.
And even within that, research suggests there are different pathways. One major pathway, often called the what pathway, seems specialized for identifying objects, figuring out what you're looking at. Another, the where-how pathway, deals more with spatial location and guiding your interactions with objects where things are and how to reach for them, for example.
Wow. Specialized streams for what and where how. That's efficient. Okay, let's circle back to color for a second. The sources mentioned two big theories, trichromatic and opponent process. How do those work? They sound a bit contradictory.
They can seem that way at first glance, but they actually describe different stages of color processing. The trichromatic theory is pretty straightforward. It proposes we have 3 types of cone cells in our retinas.
3 types?
Yeah, each type is most sensitive to a different range of wavelengths, broadly corresponding to red, green, and blue light.
Like the primary colors of light.
Exactly. And the idea is that our perception of all colors comes from the brain compared to the relative activity levels of these 3 cone types. It's like mixing those 3 colors to get everything else.
Okay, that makes intuitive sense. 3 basic inputs create the full spectrum. So what about the opponent process theory? What does that add?
The opponent process theory suggests that further along in the visual system, past the cones, color information is processed in opposing pairs. So you have cells or systems that are excited by, say, red, but inhibited by green, or excited by blue and inhibited by yellow, and then there's a black-white channel too.
Opposing pairs. Red versus green, blue versus yellow. What does that explain?
It explains things that trichromatic theory doesn't easily account for, like why we don't perceive colors like reddish-green or yellowish-blue. Those pairs oppose each other. And crucially, it explains after images.
Ah, like when you stare at a bright color and then look away.
Exactly. Stare intensely at a green square, then look at a white wall, you'll see a reddish after image. That's thought to be the opponent cells rebounding after being inhibited or fatigued.
Okay, I see. So how can both theories be right?
Because they operate at different levels. Trichromatic theory seems to be correct at the level of the photoreceptors, the cones themselves in the retina.
Yeah.
You do have those 3 cone types. But then the signals from those cones are processed by later neurons in the retina and brain in a way that sets up these opponent pairings. So both are correct, just describing different stages of the journey.
That clears it up. Different mechanisms at different points in the system.
Very cool. Okay, another huge part of vision, depth perception. Seeing in 3D, how do we manage that from the flat images on our retinas?
Great question. We rely on a combination of cues. Some require both eyes working together. Those are binocular cues. Others work even with just one eye monocular cues.
Binocular first. What's the main one?
The big one is binocular disparity. Because our eyes are set a few inches apart, each eye gets a slightly different view of the world.
Right, you can see that if you hold your finger up and close one eye, then the other, the finger jumps.
Exactly that. Your brain takes those 2 slightly different images and compares the disparity, the difference between them, to calculate depth. The bigger the disparity, generally the closer the object. This is the principle behind 3D movies, too. Ah, the glasses make sure each eye sees a slightly different image. Clever. What about monocular cues? How do we see depth in photos or paintings?
We use a whole bunch of monocular cues, sometimes called pictorial cues because artists use them. One is linear perspective. Think of parallel lines like roads or railway tracks appearing to converge in the distance.
Yeah, they look like they meet at the horizon.
Another is interposition or overlap. If one object partially blocks the view of another, We assume the blocking object is closer.
Makes sense.
Then there's relative size. If we know two objects are roughly the same actual size, the one that looks smaller in our vision is perceived as farther away. And also things like texture, gradient textures, look coarser up close and finer farther away, and position relative to the horizon. Objects higher up in the visual field often look farther away.
Wow, lots of subtle hints our brain uses automatically.
Absolutely. We piece together depth from all these clues.
The sources mentioned a really striking case, didn't they? Someone who was stereoblind, couldn't use binocular cues, but then suddenly gained depth perception.
Oh, the Bruce Bridgman case. Yes, truly remarkable. He'd been effectively blind to binocular depth his whole life, nearly 70 years. Then he went to see a 3D movie, and suddenly he started perceiving depth. The movie seemed to trigger something, and amazingly, the effect lasted even after he left the theater.
Whoa! How is that even possible?
The hypothesis is that maybe he had a brief period of normal binocular vision very early in life, allowing the basic neural circuits to form, but they then became dormant. The intense stimulation of the 3D movie might have somehow reactivated those latent pathways. It really speaks to the brain's plasticity.
Incredible story. Okay, that's a deep dive into vision. Let's switch senses now. How about hearing? How do we process sound?
Right, sound. Like light, sound travels in waves. And again, the physical properties of the wave relate directly to our perception.
Okay, so what are the key properties for sound?
Frequency and amplitude. Frequency refers to how many wave cycles pass a point per second, measured in hertz or hertz. Frequency primarily determines the pitch we hear.
So high frequency.
High frequency means a high-pitched sound, like the piccolo. Low frequency means a low-pitched sound, like a tuba.
Got it. Frequency is pitch. What's amplitude?
Amplitude is the intensity or height of the sound wave. That corresponds to the loudness we perceive, usually measured in decibels or dB.
Higher amplitude, louder sound. Makes sense.
Exactly. A whisper is low amplitude, a shout is high amplitude.
What's the range of frequencies humans can typically hear?
The general range is about 20 hertz on the low end, up to 20,000 hertz or 20 kilohertz on the high end, though we lose sensitivity, especially at the high end as we age.
20 to 20,000 hertz. And that varies for other animals too, like with vision.
Oh, definitely. Dogs, for example, can hear much higher frequencies than we can, hence the silent dog whistles.
Yeah.
Bats and dolphins go way, way higher into ultrasonic ranges.
Right. Okay, back to loudness decibels. The sources mentioned potential for hearing damage.
Yes, the decibel scale is logarithmic, meaning a relatively small increase in dB represents a large increase in sound intensity. Normal conversation is maybe around 60 dB. A noisy restaurant might be 80, 85 dB.
And loud concerts.
Can easily hit 110, 120 dBs or even more. Prolonged exposure to sounds above about 85 dBs can start to cause permanent hearing damage. And very loud sounds, say above 130 dB, can cause immediate pain and damage.
Yikes. Good reminder to use earplugs at concerts.
Definitely recommended. It's also interesting that our perception of loudness isn't just about decibels. It interacts a bit with frequency, too. We're most sensitive to frequencies in the range of human speech.
Okay, so how does the ear actually capture these sound waves and turn them into something the brain understands? What's the mechanism?
It's a pretty amazing mechanical and biological system. It starts with the outer ear. That includes the visible, fleshy part.
The bit we pierce.
That's the one. Its shape helps funnel sound waves into the auditory canal, or ear canal. At the end of that canal is the eardrum, or tympanic membrane.
The eardrum vibrates when sound hits it, right?
Exactly. Sound waves make the eardrum vibrate. Those vibrations are then transferred to the middle ear.
And the middle ear has those tiny bones.
Yes. 3 tiny bones. The ossicles, the malleus or hammer, the incus or anvil, and the stapes or stirrup. They're the smallest bones in the human body.
Wow, what do they do?
They act like a lever system. They amplify the vibrations from the larger eardrum and transmit them to a much smaller membrane called the oval window, which is the gateway to the inner ear.
So they boost the signal.
They boost the force of the vibrations, yes, which is needed because the inner ear is filled with fluid, and it takes more force to move fluid than air.
Okay, so the stapes pushes on the oval window. Then we're in the inner ear. What's in there?
The main hearing organ in the inner ear is the cochlea. It's a spiral-shaped structure, looks like a snail shell.
Cochlea. Yeah.
Inside the cochlea is fluid and a structure called the basilar membrane. Sitting on the basilar membrane are thousands of tiny sensory cells called hair cells. These are the actual receptors for hearing.
Hair cells? Like actual hair?
Well, they have tiny hair-like projections called stereocilia on top. When the stapes vibrates the oval window, it creates pressure waves in the cochlear fluid. These waves travel through the cochlea and cause the basilar membrane to ripple or move.
Okay.
And if the basilar membrane moves, it bends the stereocilia on the hair cells. This bending action is what triggers the hair cells to send an electrical signal.
Ah, the bending is the key. That's the transduction step for hearing.
That's the transduction step, exactly. The mechanical bending is converted into a neural impulse. These impulses then travel along the auditory nerve, which carries the signals out of the cochlea towards the brain.
And where in the brain does it go?
It goes through several relay stations, the inferior colliculus, the thalamus, before finally reaching the auditory cortex, which is located in the temporal lobe on the side of your head. That's where the signals are interpreted as sound. And like vision, there seem to be pathways for figuring out what the sound is and where it's coming from.
What an incredible chain reaction. From airwave to eardrum vibration, bone movement, fluid waves, hair bending, and finally, brain signals.
It's a beautiful piece of biological engineering.
Okay, so how do we tell the difference between a high note and a low note? Pitch perception. The sources mention two theories again, temporal and place.
Yes, similar to color vision, there are complementary theories for pitch. Temporal theory suggests that the frequency of a sound is encoded by the rate at which the auditory nerve fibers fire.
So faster firing for higher pitch.
That's the idea. A 100 hertz sound wave would cause neurons to fire 100 times per second, a 1,000 hertz sound 1,000 times per second, and so on.
But can neurons fire that fast, especially up to 20,000 times a second?
That's the limitation. Individual neurons can't fire that rapidly. There's a refractory period after each firing. So temporal theory works well for lower frequencies, maybe up to a few thousand hertz, perhaps using combined firing patterns across groups of neurons, but it can't explain the full range on its own.
Okay, so what does place theory offer?
Place theory proposes that different locations along the basal membrane inside the cochlea are maximally sensitive to different frequencies.
Like different spots vibrate more depending on the pitch?
Exactly. The base of the cochlea, near the oval window, is narrower and stiffer, and it responds best to high frequencies. The apex, the tip of the spiral, is wider and more flexible, and it responds best to low frequencies.
So the brain knows the pitch based on which hair cells are sending the strongest signal, like a piano keyboard laid out along the basilar membrane.
That's a great analogy, yes.
Yeah.
The place of maximum vibration tells the brain the pitch.
So do these work together, temporal and place?
Yes. The current thinking is that both contribute. For low to mid-range frequencies, say up to around 4,000 hertz, the brain likely uses both timing information, temporal cues, and location information, place cues. For frequencies higher than that, place cues seem to be the primary mechanism.
Okay, that makes sense. Combining strategies. Now, how about figuring out where a sound is coming from? Sound localization.
Also super important for survival. We use two main types of cues here. Monaural cues, using just one ear, and binaural cues, comparing information from both ears.
Monaural, how can one ear tell direction?
Mostly through the shape of your pinna, the outer ear. The way sound waves bounce off the folds and curves before entering your ear canal changes slightly depending on whether the sound is coming from above, below, in front, or behind you. Your brain learns to interpret these subtle changes.
Huh. So the shape of my ear actually matters for localization.
It does, especially for vertical localization. But for figuring out left versus right, the horizontal plane, binaural cues are key.
Okay, binaural means using both ears. What are those cues?
There are two main ones. First, interval level difference, ILD. If a sound comes from your left, it will be slightly louder in your left ear than your right ear because your head creates a sound shadow, blocking some of the sound from reaching the far ear.
Okay, difference in loudness. What's the second one?
Interval timing difference, ITD. That same sound coming from your left will also arrive at your left ear a fraction of a second before it arrives at your right ear.
A tiny time difference.
Tiny, but detectable. Our brains are incredibly sensitive to these minute differences in both level and timing between the two ears, and they use that information to calculate the horizontal position of the sound source with remarkable accuracy.
Amazing how much computation is happening just to locate a sound. Now, unfortunately, hearing doesn't always work perfectly. The sources mentioned hearing loss. What are the main kinds?
Broadly, there are two main types. Conductive hearing loss happens when there's a problem in the outer or middle ear that prevents sound from being conducted effectively to the inner ear.
Like what? Earwax?
Yeah, earwax buildup is a common one. Or fluid in the middle ear, an infection, a punctured eardrum, or problems with those little ossicle domes. Often, conductive hearing loss can be treated medically or surgically or helped with conventional hearing aids that just amplify the sound.
Okay, so conductive is about getting the sound to the inner ear. What's the other type?
Sensorineural hearing loss. This involves damage to the inner ear itself, specifically the hair cells in the cochlea, or damage to the auditory nerve that carries the signal from the cochlea to the brain.
And what causes that?
Lots of things. Aging is a big one. Genetic factors, exposure to loud noise over time, that's a major preventable cause. Certain illnesses, some medications, head injuries.
And this type is harder to fix.
Generally, yes, because the sensory receptors or the nerve pathway is damaged. Hearing aids can still help sometimes by making sounds louder, but if the damage is severe, they might not be enough. That's where cochlear implants come in.
How do those work?
A cochlear implant bypasses the damaged hair cells entirely. It has an external part that picks up sound and an internal part implanted surgically, with electrodes threaded directly into the cochlea to stimulate the auditory nerve fibers electronically. It doesn't restore normal hearing, but it can give a sensation of sound and allow many people to understand speech again.
A direct electrical stimulation. Fascinating. The sources also briefly mentioned Meniere's disease.
Yes, that's a disorder of the inner ear that affects both hearing and balance. It causes episodes of vertigo, tinnitus, ringing in the ears, a feeling of fullness, and fluctuating hearing loss, which can sometimes become permanent over time.
Sounds really disruptive. Okay, we've covered light, sound. Let's move to the chemical senses. Taste and smell.
Right. Gustation, taste, and olfaction, smell. They're called chemical senses because they respond directly to molecules in our food, drink, and the air we breathe.
And they work together really closely, don't they, for flavor?
Absolutely critical. What we commonly call the taste of food is actually a combination of taste from the tongue and, very importantly, smell from the nose. Think about when you have a bad cold and everything tastes bland.
Yeah, food just isn't the same.
That's because your sense of smell is blocked, and you're missing a huge component of flavor perception. The aroma molecules from food travel up the back of your throat to your nasal cavity while you chew.
So flavor is really taste plus smell. Okay, let's focus on taste first. The old idea was just 4 basic tastes. Sweet, sour, salty, bitter. Is that still right?
Mostly, but it's been expanded. Those 4 are definitely key, but we now widely recognize a 5th basic taste: umami.
Umami. What's that like?
It's often described as savory, meaty, or brothy. It's triggered by glutamate, an amino acid found in things like Parmesan cheese, mushrooms, soy sauce, and MSG.
Oh, that savory depth.
Okay. Anything else?
There's also growing evidence for a 6th basic taste receptor, specifically for detecting fatty acids. The taste of fat.
A taste for fat. That makes evolutionary sense, maybe. High energy source.
Potentially, yeah. So at least 6 basic taste categories now seem likely.
And how do we detect these? Where are the receptors?
The receptors are located in taste buds, and taste buds aren't just on the tongue. They're also found on the roof of your mouth, your throat, even your epiglottis.
All over the place.
Yeah, and these taste buds contain clusters of taste receptor cells. Each cell has little extensions that poke into a taste pore where they can interact with dissolved molecules from food. Interestingly, these taste cells have a short lifespan, only about 10 days to 2 weeks before they're replaced.
Wow, constant turnover. Keeps the system fresh, I guess. When a food molecule binds to a receptor, it triggers a signal in that cell, which then gets sent via cranial nerves towards the brain.
And where does the taste information end up?
It travels first to the brainstem, medulla, then it gets relayed through the thalamus and onto the gustatory cortex, which is in the frontal lobe near where it meets the temporal lobe. Some signals also go to the limbic system, which is involved in emotion and memory.
Explains why some tastes can feel comforting or disgusting, maybe. That limbic connection.
Could be part of it, yes.
Okay, now smell. Olfaction. How does that work? Less direct contact than taste.
Right. For smell, the receptors are located high up inside your nasal cavity in a little patch of tissue called the olfactory epithelium.
Way up in the nose.
Yep. Embedded in the mucus lining, there are millions of olfactory receptor cells. Each one has tiny hair-like cilia extending into the mucus. Airborne odor molecules that we inhale dissolve in this mucus and bind to specific receptor proteins on these cilia.
So molecule binds to receptor hair, then what?
That binding triggers a signal within the receptor cell, which then sends an impulse directly up to the olfactory bulb.
Olfactory bulb, where's that?
It's actually a structure at the very tip of the frontal lobe of the brain, sitting right above the nasal cavity. It's like the brain reaches down to meet the nose.
A direct connection.
Very direct. From the olfactory bulb, signals travel to other brain areas, including, importantly, parts of the limbic system again, emotion and memory centers, like the amygdala and hippocampus, and also to the primary olfactory cortex, which is located near the gustatory cortex.
That close link to the limbic system must be why smells can trigger such strong memories and emotions, right? Like Proust's Madeline.
Exactly. It's a much more direct route to emotional centers than some other senses have.
And like hearing and vision, smell sensitivity varies across